Yeah. Okay. So our next talk of this session is given by Amber Smith. So she's from the University of Tennessee at Memphis, and okay, so she will talk about virtual patient cohorts and how they reveal the immune mechanism for the outcome of the disease. So please start.
Thank you so much, and thank you for the invitation to talk today and talk about this work and how we approached a problem of really looking at human heterogeneity and the variety in disease outcome. I'll start with acknowledging all the people that did the work. This was a really fun collaboration with Morgan Craig's group at the University of Montreal, and Adrian Jenner really led quite a bit of the work here. And then there was a handful of people from my lab that contributed, Penny Morell at the University of Pittsburgh and Courtney Davis at Pepperdine University. So I don't have to give obviously too much of an introduction into COVID-19, and Miriam just did a really great job of highlighting, some of the challenges that we've had so far, and while we're actually still in the pandemic. Although if you ask some of our governors here in the US, they might tell you that the pandemic is over. And that's in part because our cases are declining, but they're pretty steady actually right now. And of course we have the B117 variant that's more transmissible, and we're starting to get some travel related cases of other variants that we're obviously very concerned about. Currently, at least in the US, we've had about 32 million cases and 575,000 deaths. And that's pretty robust. And people like to compare it to past influenza pandemics. Obviously, this is significantly less than maybe the 1918, although it's approaching that, but is maybe almost double the size of the 2009 pandemic that we saw. The US is maybe, from what I've heard from some of the others on the call today, that we're maybe a tiny bit ahead in our vaccinations, although it's kind of flattening out for the number of people that actually want to be vaccinated. So we're really trying to put together some vaccine campaigns to address the vaccine hesitancy. But to date, we've had 247 million vaccines administered. And that really, I think, drove that decline that we saw between January and March. I put a graph up here of the age distribution of the cases. So as you can see in this figure, the highest number of cases has actually been in the 20 to maybe 30-year-old range. That's obviously completely different than what we see in the more severe disease, which predominantly affects the older generation. And then thankfully, the children have not been quite as affected, although there are still quite a few cases in those individuals. So one of the things that we've learned over the past year is that the immune response to SARS-CoV-2 is challenging to understand, and that there's a variety of outcomes, but also a variety of host responses and other clinical manifestations. So what I'm going to talk about today, and the model that we developed, really focuses on this immune response and trying to understand some of that heterogeneity. So for those of you who might not be familiar, I'll just give a very brief kind of basic overview of immunology. When viruses come in and infect cells, as you see here, those cells are going to secrete interferon, which can put other cells in an antiviral state. The cells, as well as the resident macrophages, will call in help, and they'll secrete other cytokines. These are a handful of them here that bring in the T cells, your monocytes, macrophages, neutrophils, that come in and target the virus, but they also target the infected cells and hope to resolve the infection. There's quite a lot of very complex dynamics that happen. Of course, early on, you have your alveolar macrophages that are trying to clear it up. They often lose their battle pretty quickly, but are a big initiator of the host response. They bring in the macrophages and the neutrophils, and then ultimately the adaptive response comes in, where you have your CD4 T cells and sort of your usual gold star of an immune cell is a CD8 T cell that can kill the infected cells and clear out the infection. So in a healthy immune response, you get a very robust CD8 response that clears the virus out, and then you'll generate antibodies so that you don't get infected again. And all of these things then work to try to manage the inflammation and the amount of lung damage. Now with COVID-19, the immune response is pretty dysfunctional overall. So there's an excessive infiltration of macrophages, monocytes, and in some cases T cells, although there's definitely data that shows that T cells can become depleted, and that is a driver of the severity of COVID-19. And then of course, there's a huge systemic cytokine, what some people call a cytokine storm. Those cytokines themselves are not exactly causing all of the inflammation, or directly, but they do cause cells to come in. Those cells then create a lot of inflammation. You get pulmonary edema, pneumonia, and then there's other effects on other organs, such as the heart that we've seen, as well as the blood vessels. So it's not just a lung disease, which has made this obviously very difficult to treat, in addition to trying to just manage the symptoms. So there's been, over the last year, quite a bit of work done in human studies to look at various host responses. And what has come out of most of these studies is that host responses are incredibly variable for probably a variety of reasons, which I'll talk about in a few minutes. But there are some consistencies across a handful of different studies that have looked at these factors. One of them is that more severe patients pretty consistently have an elevated IL-6. So IL-6 is one of your cytokines that really brings in your macrophages. In all of these studies, you see this kind of stepwise increase from when you go from somebody who's healthy to someone with a moderate disease to someone with a severe disease. It just keeps going up and up and up. The other thing that's been fairly consistent is that your CD8 T cells are actually depleted. So you need these cells there to come in and clear out the infected cells. So if they're not there doing their job, then the infection gets to persist. And that's been seen across a handful of different studies too, where you should hopefully have quite a few CD8 T cells, you get this depleted amount and persistence of the infection. And both of these things have also been shown within mouse models, and they were actually all shown prior to this entire pandemic for studies that were done on this SARS-CoV-1 virus that infected quite a few people back in 2002 and 2003. So there are some really consistent things across some of these SARS viruses, and of course there are some differences as well. So one of the other things that was shown in the same type of dynamics that we saw with the IL-6 is there's elevated interferon levels. So this was a study looking at healthy versus non-critically ill versus critically ill. And you can see that some of these cytokines have distinct differences. GM-CSF was another one that was consistent across studies. GM-CSF is responsible, it's a chemotactant that brings in your macrophages. And in this particular study up here on the right, they actually looked across different severity levels as defined by the WHO. And you can see this sort of stepwise increase of GM-CSF. And that was the same behavior that we saw with IL-6 as well. And in yet another study, they took a separate approach and actually looked at some of the lungs of infected patients and did some really interesting studies where they were trying to understand what is actually happening in the lung and in different areas of the lung. So in both of these figures, the blue, you have higher amounts of cells where the virus is actually lower. And then where the virus is higher, you have fewer cells there. So that could be indicative of various things that are happening. One is that the reason why the cells are there and in higher numbers is because they're clearing out that virus, which is why you find lower virus. But there, of course, could be other aspects to that. And this is cells per millimeter squared. So you can see also there is some variation to that. And they said in their work that there was also a lot of spatial heterogeneity. So these are essentially human lung slices, although they're color coded with where they found the cells and where they found the virus. So the same with this upper right-hand figure. The red is areas that are SARS-CoV-2 positive, and the blue are areas where there's SARS-CoV-2 negative. And they looked at the monocytes, macrophages, and T cells, and they had a couple other cell types in there too, but for the purposes of today, I left those particular ones out. But even across each of these columns is a different patient. So you can see that even within different patients and within different areas of the lung, they have completely different responses. Now, how much the local response in these areas drives the more global response is sort of unclear. It may not matter at all, or it could be one of the really big drivers of the heterogeneous responses that we actually see. There we go. So in the same study, they also looked at just a PCA analysis of those that were critically ill versus non-critically ill versus the healthy, and listed here out the days post-infection. So you can see actually that they group, but they group pretty broadly depending on the patient. And there could be several contributing factors to the heterogeneity that we're seeing. One is definitely age. That figure that I showed you earlier, shows who gets predominantly infected, but those are also the people that are not necessarily more severe clinical manifestations. There are sex differences. There's of course genetic differences, both from the host and various aspects of the immune response, but also the virus. And like I mentioned earlier, all the variants that have come up, they do initiate at least some slightly different immunological responses. And then that B117 variant infects cells more readily, and of course then can transmit more readily. Anybody with an immunologic defect, and I don't mean that as an immunocompromised, because actually the immunocompromised, there's not a lot of evidence that they're disproportionately affected. But there has been other studies that have shown that patients with antibodies to interferons fare worse. So you actually need that interferon response, but there are some people that have antibodies to that. And of course, dose will play a huge role. Or could play a huge role. The transmission route, whether people are predominantly getting infected within your upper respiratory tract that then migrates down to the lower versus say inhaling a big dose straight into the lungs could drive differences here. But then we also run into just a data heterogeneity problem where the day of sampling probably matters a lot. And what a lot of clinical studies do is they try to go back to when the onset of symptoms started. But as we've probably heard a lot over the last year, the symptoms vary quite a bit. Patients may or may not be able to identify that date better. So we might just get natural heterogeneity from just sampling, say, on one time point or on one or two time points when we don't actually know if those are the same time points across patients. So there's a lot of heterogeneity that can go into this data, which obviously makes understanding it and picking out the important drivers or the biomarkers that might say why a patient goes one way versus the other really difficult to tease apart. And so that's where we came in with our mathematical approaches to really try to dissect that immune heterogeneity. So our approach to this was first to develop and calibrate a comprehensive model. Now, I'm a huge fan of small models that are really tractable, and we can know everything about them and how they behave and know exactly what's going in, and enough to explain a piece of data. Unfortunately, those small models are often limited in what they can assess. You still can put in heterogeneity into those, but if you're really trying to look at various aspects of the host response, you of course need to model more aspects of that host response. And so we developed a really comprehensive model that I'll kind of walk you through over the next couple slides that we also tried to then reduce the parameters, because with a larger model, of course, you have many parameters that you need to calibrate. So we did that through sub-models and through various pieces of data that were, some were COVID related, some were not COVID related, so that we could try to really parameterize our model without doing just a traditional blanket fit all at once. And then we did a sensitivity analysis to determine what were the drivers of a mild versus severe disease. We did a little bit of work looking at whether or not, what happened if we knocked out a response. And then we used these ideas to construct a virtual cohort to actually look at the heterogeneity within a population, or what could drive the heterogeneity within a population. So when we started sort of back at the beginning. This is a schematic of a standard viral kinetic model, and a handful of papers already in the literature have used this exact type of model to actually fit these exact data. These have shown up in a handful of places because they have temporal data where they're from patients in Singapore and Germany. So you can see a lot of time points, which is always what we look for as mathematicians, and various dynamics between the different patients. So within this model, we have our susceptible cells that become infected with virus. Those can then die off because the virus kills it, but they can also produce more virus that of course can infect more cells. And you can see the fit here to all of the data as well as the individual fits. And so the model does a pretty good job. And if that's all we were doing was looking at viral loads, we could sort of be done here. And that's what some of these other studies have done as well. Because to replicate viral loads, we actually don't need to incorporate a lot of immunological dynamics. They're really kind of wrapped up in these parameters. And then we took the next step and looked at the tissue, just the tissue response. So not more of a systemic immune response, but interferon gets produced when a cell gets infected. And when that happens, those cells then can become resistant to infection. And these models have been in the literature for a long time as well, but we wanted to just kind of build our model piece by piece to really get a sense for what was happening, and to really look at what some of the data, both clinically and in the past animal studies with SARS-CoV-1, they all have suggested and showed that a delay in the interferon response can really change the dynamics of the infection. And so that's what you can see here in these figures. So where the delay is in the dashed lines, and then the situation where there is no interferon delay is in the solid lines. And so if you have that delay, of course, you might get higher viral loads. You'll get more of your cells becoming infected because not as many are becoming resistant. Okay, so then the challenging part actually comes with the rest of the immunology. So getting the dynamics of different cell populations, the macrophages, the neutrophils, the CD8 T cells coming in, and then the cytokines also, GM-CSF, IL-6, G-CSF, and there's a lot of complicated dynamics that happen within the immune response, and there's a lot of feedbacks that happen. And so our approach to building the model for these was to take data from the literature that we could find that either had dose responses, such as this one in the middle, where across the X is IL-6 at different doses, and the response of, say, a CD4 T cell on the Y. And then we fit a curve to it. Most of these curves really look like a Hill type function or a Michaelis-Menten type of function to get that dose response. And then we incorporated that into the model. So on the left-hand side, this is actually production of IL-6 from monocytes. So that allowed us to look and acquire a production rate for that particular cytokine from those cells. And then on the right-hand side, this is just monocyte production. And so we sort of scoured the literature to try to find as much data that we could to calibrate each aspect of our model so that we could put it all together. And I won't show all the equations because there's too many that could probably fit on the screen, and we could talk about the development of the model for probably a very long time. So I just put up the full model schematic. Over here on the left-hand side is really the models I just showed you, where we have virus that infects cells. Those cells can become resistant via interferon. And then all of the immune response dynamics. So here you have your neutrophils coming in because you have cytokines being produced. You have macrophages coming in and regulating the different types and the activation of those macrophages. Your monocytes coming in and becoming macrophages. And then of course the T cell response. So this is what some of the dynamics from this model look like, and what they look like for a severe case, which is in these dashed lines, versus a mild case. So you can see the viral loads that I showed you earlier here, and what a model might suggest. And we can talk about what the definition of mild versus severe is, because that's also a little bit complicated, but let's just say maybe better or worse. So with the severe disease, you actually clear out virus a tiny bit faster, and that is seen in both, it's at least seen in animal studies. I'm not sure that anybody has shown it robustly in humans yet, but this is the behavior that we see within animal studies. In the more severe cases, you would of course get more infected cells. That leads to more inflammatory macrophages. We were able to recover the depletion of CD8 T cells, and then the increase in IL-6. And if we look at a couple others, with interferon, we actually, in the more severe case, recovered the delay that is seen in patients that do worse during their COVID infection, a higher amount of GM-CSF, which I mentioned earlier, we also see within patients, and then a higher level of neutrophils. So most of the things that our model produced are actually seen clinically, and that was not all, say, built into the model. A lot of that happened actually really naturally by putting in the dynamics, say, of all those cells and cytokines that were calibrated to other data that should really show the dose response. So of course, you know, people can probably do a lot with a large model, but I will tell you that a lot of what we found was just naturally produced by getting those dynamics right. So once we had a model that we felt pretty good about, we decided to then develop a virtual cohort out of it so that we could put in that type of heterogeneity that might be seen within, say, a patient cohort if you were to do a clinical trial. And the way we generated it was we first drew parameters from a normal distribution. And we only did select parameters that we already knew were more sensitive, because of course, if you have a non-sensitive parameter, you're not going to get different dynamics. So we drew only a handful of different parameters. Then we would evaluate the model, make sure that we were still within the physiological ranges that have been seen in data. Here's two examples over here. We only did it with a handful of data that was available at the time that we generated this cohort. And then once they were in those physiological ranges, then we could, say, accept them into our cohort. And this is sort of what some of the cohort dynamics looked like, and the viral load that we used to constrain it within that. So here's our viral load dynamics, our IL-6 dynamics compared to the data. So for some of them, we actually do get the full spectrum. For other ones, that we got pretty close to the mean, but maybe didn't get all the heterogeneity. And this was all just naturally from generating that cohort. But say with like monocytes weren't different between the moderate and severe, and the same with the neutrophils, but we did capture the increase in IL-6, which of course is over here. And on the right-hand side, this is just to remember, say, what those IL-6 dynamics looked like from a severe patient compared to a mild patient. So once we had this, we wanted to take it a step further and actually assess inflammation. And our knowledge of how to potentially assess inflammation in part came from some work that my lab has done on influenza. So this is a preprint that we have out right now where we looked at the infection within the lung. So up here on the right-hand side is a slice of a mouse lung, and the colored in parts are where are positive for the virus. And so that's the amount of the lung that is infected, say 3 days into the influenza infection. And then we applied a model and did a variety of analyses, and we ultimately ended up showing that these, what are called lesions, correlated pretty nicely, but also non-linearly to weight loss in mice, which is their measure of disease severity. And then we also had a measure from a pathologist of looking at inflammation. Inflammation and disease severity are also correlated. These are slightly different, but they still have that non-linearity to them. And then inflammation is actually correlated fairly linearly to both macrophages and neutrophils. And then I didn't show here, but IL-6 follows pretty close to macrophage dynamics as well. And so we developed this inflammation biomarker to try to assess in our cohort who might be inflamed and who might not. And so what we did was we had a combination of the IL-6, of the neutrophils, and of the damaged lung tissue, which is essentially what you would see something like here on this right-hand side. So by doing that, and then across the x-axis, we actually ordered the patients by their inflammation biomarker to where you have less severe on the left-hand side, and as you sort of follow up this green curve, you increase in severity. So unsurprisingly, as IL-6 increases, severity also increases. But as you notice here in our bioinflammation, or in our inflammation biomarker, we had neutrophils in there. Neutrophils essentially came out of it as not being directly connected to whether or not somebody might be more severe or have higher inflammation. So the macrophages were also increasing, but say not earlier on. We looked at interferon exposure, so like the area under the curve of interferon, and it was not correlated whatsoever. The peak time of interferon, though, was, but it had some interesting dynamics, and then it's almost a bifurcation, where the first half of our cohort, which does have increasing severity, had essentially the same or no peak, and but once you sort of hit a critical level, then you jump up to having a higher peak in interferon, and that's in your more severe disease. And then, of course, with the T cells were also not in our inflammation biomarker, so it just naturally fell out. That as your T cells decline, your severity goes up. And all of these things, for the data that is available, either from animal studies or from humans, were pretty consistent. So then we took it one step further, because we don't think it's quite enough just to say, oh, if your T cells are low, then you're going to have a worse disease, because it's a little bit more complicated than that. And we're also interested in what's driving those dynamics. And so then what we looked at was the parameters that were driving some of these things, and whether or not those parameters themselves were, say, correlated with some of the other ones that were also driving that inflammation. So here on the bottom left, monocyte to macrophage differentiation rate, which is mediated by IL-6, but it's the rate itself, was really highly correlated to this interferon peak. And those two things are completely separate dynamics within the model. So we thought that was really interesting, that you kind of get this type of split. And that bifurcation that we saw in the peak of interferon is still visible here, where you have a whole bunch that are low, and then that once you sort of hit that critical level, you get more severe disease, but this is still pretty linearly correlated across there. And then we also looked at the production rate of interferon and the peak, and you can see that split again here, but not so much of a correlation between the two. And then finally, we looked at sort of it's in a 2-dimension, but it was really a 3-dimensional type of plot and information, where we looked at IL-6 versus interferon versus T cells. So the maximum amount of interferon is on the x-axis, the maximum of IL-6 is on the y, and then the colors of these dots are the amount of your T cells, of the maximum level of your T cells. And you can also see that here, when you have low T cells, you have high amounts of IL-6. So that was really correlated to that, comparatively to your max interferon, which is not necessarily quite as correlated. So I will just take a couple seconds to summarize what we have done. So we were really interested in looking at the heterogeneous responses and the heterogeneity in outcome and what might actually be driving that. So we developed this virtual patient cohort to try to answer that question, or at least provide some insight into it. What we found is that we had a model that really reproduced quite a bit of the behavior that's seen both clinically and experimentally. The model was able to produce most of the dynamics. It was able to recover some of the features of coronavirus disease, including the delay in interferon peak, a high IL-6 and low T cells, and that correlates with disease severity. And then on top of it, the mathematics added to that a little bit and suggested that it's a high rate of differentiation from monocytes to macrophages that actually mediates that. So it's not just that it's there, it's actually the rate at which it's happening. And then of course that was mediated by IL-6 and having the reduced interferon then. And then that sufficiently delayed interferon production did lead to higher amounts of inflammation, and that high IL-6 ultimately led to the CD8 T cell depletion, although not directly, because IL-6 has a little bit different of a function. So I will stop there and take any questions that you might have.
Right. Thank you, Amber, for your talk. Is there any questions? Or you can raise hand, or maybe send something to the chat if you have a question. So in the meantime, I wanted to ask, out of curiosity, so you have so what you have built is a model for inside the host propagation of the disease in a way.
Yes.
And with a high focus on the immune response, and the way we can get rid of the disease in a way.
Yeah. Maybe
the time it takes to get rid of it. Do you think you could get some convincing estimates of the viral load? Well, what I have in mind is try to quantify or to predict in a way the transmissibility of a particular host. Well, given what we know.
I think the short answer is yes. So what we know from animal studies in other viruses is a couple of things. One is that most of the transmission is correlated and really non-linearly correlated to the amount of virus that's in the upper respiratory tract. And that of course makes a lot of sense, right? If you have a lot of virus in your nasopharynx and you happen to sneeze, or if you're talking, then of course you're going to spew out a higher dose, and that's going to lead to higher transmission. What I think we're not 100% certain of, or that I've seen any data for yet, but there is some data with influenza in animal models, is that a lot of the transmission actually happens before the onset of symptoms, or right up until when that virus is really replicating a lot and you're getting up to that peak and high titers, but your immune response hasn't quite come in yet to start taking care of it. And so I don't know if that happens, say across the board with respiratory viruses, if that's just, something that is specific or not, or if that's what COVID does. But I would say that if you have a lot of virus in your upper respiratory tract, you're going to have a higher probability of transmission.
Right. Thank you. And maybe another question is, because the model is very complicated, I didn't quite get it. So how does the virus move in the body?
We don't have any spatial aspect to it. So as far as like physical movement of the virus, everything is considered a well-mixed system. So we use ODEs, and there's no PDEs within this at all. And that, at least from work in other viruses, and particularly our work with influenza, it hasn't inhibited any of our conclusions that we have been able to make. But there obviously is some spatiality to the infection, and probably to even some of the other aspects of the disease. You know, if you get into the lower areas of the lung, you're probably going to have potentially more severe disease.
Okay, so that means that in your model, you consider that every cell responds the same way, or on average the same way to the virus, right?
Yes, yes, we do. Yeah.
Okay, okay. So okay. That would also be quite interesting to differentiate these responses and to get more information on which tissues would be most damaged by the virus and so on.
Yep.
But,
yeah, yeah. And, you know, trying to get at that question is initiated some of our work on influenza, and that histopathology, that whole lung, we call it histomorphometry image. The whole idea behind that was to try to look at what is actually happening in the lung. You know, where is the virus in the first place? What do the dynamics, you know, across time look like? We can still get and replicate a lot of the dynamics, but there of course are other things that we can't capture, or you know, maybe some of those nonlinearities are because of that. Like, you know, we can capture some of those correlations, say between inflammation and disease severity, or the, you know, amount of those lesioned areas or infected areas compared to disease severity, but it might be nonlinear because there is something else happening there.
all right, thank you. So that answers my questions. Is there any other questions?
Can I ask one? Okay. At some point, you show a lot of curves, a lot of data for each type of cell, and is that coupled model, or is that a full model? You try to fit to all those curves?
Yeah, what we tried to do is we took submodels and, you know, portions of a model to try to either get out parameter values or functional responses. And then we put that into the larger model. So the full model is all coupled together. There's nothing that is, say, still done in the submodel and just pulled in.
So you first fit the simple model for a single.
Yeah.
And then you use that to estimate the parameter for the complex model. Is that correct?
Yeah, so we estimated the parameters that are in that smaller model, and then carried those over to, say, the next step where the model was a little bit larger, and if parameters needed adjusted, it was easier to adjust them than refit everything, because there's not, you know, the more we added, the less data there were, you know, were actually available over time. So a lot of the, you know, the clinical data that's coming out is just like a single snapshot of what's happening. So we carried those parameters in, and then if we did have data, we would, you know, estimate or fit just those particular parameters, and then sort of go to the next step.
So that's a way to, you use a simple model in order to what I call, that sort of phenomenological model, not describing the interaction, but plugging that into the complex model, you will have some information about how to estimate.
Yeah, it was really like a parameter reduction problem. Yeah.
Right. And usually, from what so it's surprising. For once, it's working.
Yeah.
For immune system, usually it doesn't work, right?
Yeah. Yes.
So any idea why this time it's working? Because maybe you take into account more cells, more type of cells, or.
Yeah, that's a great question. You know, the smaller models we had a lot of success with to investigate specific problems. You know, we still obviously have to include phenomenological things, and even in this model, there's not everything and every aspect of the host response is in there, but we tried to build enough in at least of the inflammatory responses and the most important cell types that are really important actually for every virus infection and probably every bacterial infection. And then just took a lot of care to develop that. So, and I will tell you that the model and what it produces actually looks a lot like some of our influenza data as well. So we're hoping to kind of, you know, repurpose it to start to look at some of that also, where we do have a lot of data, at least animal data. Human data is, of course, still scarce.
Okay, I see. You are using some influenza virus for animals, some sort of strain of influenza for animals to make some experiments. Is that right?
Yep. Yeah. So actually, half of my lab does experimental work. So all the data that we use,
we get a lot of data, basically, about immune system, but for animals, right?
Yes.
Oh, I see. Oh, interesting. Okay. So, and just by curiosity, what that's the first time I hear about this IL-6 cells. What's that about?
IL-6 is a cytokine. It's traditionally labeled as an inflammatory type of cytokine. So macrophages will produce it. They'll also bring in macrophages as well. So you'll see IL-6 actually in a lot of contexts. It's always pretty high in virus infections as well as bacterial infections. And that's predominantly because you have macrophages that are trying to manage the infection and keep the inflammation down, but enough cells in there to coordinate the immune response.
So apparently this is a very important
it is,
in your model, right? And
it turned out to be really important here. It has some other more challenging features. You know, people have tried to use IL-6 treatment, and it tends not to work very well. And there's various reasons, I think, why that is.
Okay, very interesting. Okay, thank you.
Okay, thank you. So I think we have to stop here because I have to go home before the curfew. So thank you again, Amber, for a very nice talk, very interesting, and thank all of the participants for their presence here today. Okay, so I don't think we have another talk planned, so this may be the last one of this series, and so okay.
So you were probably the last one. So I see Glenn, who was one of the first.
Yeah, the very first.
The very first. And so we will stop with a very nice talk here, and we are very glad about that. Thank you very much.
Yeah. Thank you, everyone, and bye-bye.
Bye-bye.